Vitamin B3, in its multiple forms, nicotinic acid, nicotinamide, nicotinamide riboside, NR, and nicotinamide mononucleotide, NMN, is increasingly being recognized for its fundamental role in maintaining DNA stability and supporting cellular mechanisms that may reduce cancer risk. While no nutrient can completely prevent cancer on its own, B3 plays a critical supporting role in the body's intrinsic defense systems by fueling pathways that repair DNA, regulate cellular metabolism, and mitigate oxidative and inflammatory damage. These mechanisms are essential because accumulated DNA damage is a key driver of cellular dysfunction, genomic instability, and ultimately tumor formation. By supplying the necessary substrates for NAD-dependent repair enzymes, vitamin B3 strengthens the body's natural capacity to maintain genomic integrity, providing a scientifically grounded strategy for long-term cellular protection. One of the central ways B3 contributes to DNA stability is through its role as a precursor for NAD, a coenzyme that powers enzymes involved in DNA repair, including PARPs, poly-ADP ribose polymerases, and sirtuins. PARPs detect and respond to single-strand DNA breaks, signaling repair machinery and coordinating the restoration of the DNA helix. When NAD levels are insufficient, PRP activity diminishes, allowing damage to accumulate and increasing the likelihood of mutations that can lead to cancerous transformation. Niacin supplementation restores NAD availability, ensuring that these repair enzymes function optimally. Laboratory studies and emerging human data indicate that enhanced NAD levels promote more efficient repair of DNA lesions, preserve chromosomal stability, and reduce markers of oxidative stress that contribute to mutagenesis. In this way, B3 operates as a metabolic first responder, equipping cells with the tools they need to maintain genomic integrity even under conditions of oxidative or environmental stress. Sirtuins, a family of NAD-dependent enzymes, provide an additional layer of protection. These proteins regulate gene expression, modulate stress responses, and coordinate metabolic adaptation, all of which influence DNA repair processes indirectly. By activating sirtuins, vitamin B3 supports the removal of damaged proteins, enhances mitochondrial function, and reduces chronic inflammation, all factors that contribute to genomic stability. This multi-layered approach is particularly important because cancer risk increases not just with direct DNA damage, but also with the cumulative effects of metabolic dysfunction, oxidative stress, and inflammatory signaling. NR and NMN supplementation have been shown in preclinical models to enhance sirtuin activity, improve mitochondrial health, and reduce oxidative DNA lesions, demonstrating that B3 provides broad cellular protection beyond a single repair pathway. B3's role in inflammation modulation further strengthens its potential protective effects. Chronic low-grade inflammation is a recognized driver of both DNA damage and cancer progression. NAD restoration supports sirtuin-mediated anti-inflammatory pathways, reducing the production of pro-inflammatory cytokines while maintaining normal immune surveillance. By controlling inflammation, B3 indirectly reduces DNA insult and maintains a cellular environment conducive to accurate repair and controlled cell proliferation. This integrated effect, DNA repair plus inflammatory regulation, helps explain why higher dietary or supplemental intake of B3 has been associated in epidemiological studies with lower incidence of certain cancers, particularly those affected by oxidative stress and chronic inflammation, such as skin, gastrointestinal, and metabolic-associated cancers. Vitamin B3 also enhances cellular energy metabolism, which is critical for effective DNA repair. Repair processes are energy-intensive, and NAD-driven mitochondrial ADP production provides the fuel required for protein synthesis, chromatin remodeling, and enzymatic repair activity. Without sufficient energy, cells may initiate partial or error-prone repair, increasing mutational risk. By ensuring adequate NAD levels, NR and NMN supplementation support mitochondrial efficiency, enabling cells to meet the high-energy demands of DNA maintenance. This dual benefit, repair machinery supported both by coenzyme availability and by cellular energy, underscores why B3 is uniquely positioned to contribute to long-term genomic stability. Safety, accessibility, and translational potential make B3 particularly compelling for preventive strategies. Niacin and nicotinamide have long histories of safe human use, while NR and NMN have demonstrated excellent tolerability in clinical studies, even at doses sufficient to elevate NAD significantly. Unlike pharmaceutical interventions that target a single pathway, B3 supplementation provides systemic support, reinforcing multiple interconnected protective mechanisms across tissues. This systemic effect is particularly important because cancer is rarely isolated to a single cell type or organ. NAD replenishment supports DNA stability and metabolic resilience in multiple organ systems simultaneously, including the liver, skin, gastrointestinal tract, and hematopoietic tissues, where mutagenic stress is particularly high. Importantly, while B3 is not a standalone solution, it complements lifestyle strategies known to reduce cancer risk, including regular exercise, balanced nutrition, avoidance of tobacco and excess alcohol, and adequate sleep. By fortifying cellular repair mechanisms, reducing oxidative stress, and modulating inflammation, NAD-boosting B3 compounds enhance the efficacy of these interventions and create a more resilient metabolic and genomic environment. Emerging research also suggests potential synergies with other anti-aging and DNA-supportive compounds, pointing to integrated strategies for long-term cellular protection. In conclusion, vitamin B3 plays a central role in DNA repair, genomic stability, and metabolic resilience, positioning it as a nutrient with promising cancer protective potential. By replenishing NAD, supporting PRP and sirtuin activity, enhancing mitochondrial energy production, and moderating inflammation, B3 strengthens the body's intrinsic defenses against DNA damage and mutagenesis. While it is not a cure or guaranteed prevention, the growing body of evidence indicates that NAD-boosting B3 compounds can provide systemic cellular support that may reduce cancer risk, slow age-related genomic instability, and enhance long-term tissue resilience. By integrating B3 supplementation into a broader lifestyle and metabolic strategy, individuals can leverage a scientifically grounded, practical, and systemic approach to maintaining DNA integrity and supporting long-term health span.